The cell cycle is a fundamental process that drives the growth and reproduction of all living organisms. Growth is one of the most defining characteristics of living organisms. From the smallest single-celled bacteria to the largest multicellular creatures, all living beings grow by increasing in size, adding new cells or both. But how does this growth happen? The answer lies in the cell cycle, a vital process that ensures organisms can grow, repair damaged tissues, and reproduce. Today we'll explore the importance of the cell cycle, the stages involved, and how DNA replication plays a central role in this process. Let's start with the basics. The cell cycle is a series of carefully regulated steps that a cell undergoes to grow, prepare for division, and ultimately divide into two identical daughter cells. This cycle is essential for the growth and maintenance of all multicellular organisms. Without it, plants wouldn't grow taller, animals couldn't heal wounds, and no living organism could replace aging cells. The cell cycle is divided into two main phases, interphase and the mitotic phase or M phase. Interphase is where the cell spends the majority of its life. During this time it grows, performs its specific functions, and prepares for division. Interphase itself is divided into three stages, G1, S, and G2. The first stage, G1 or GAP1, is a period of growth. During G1 the cell increases in size, synthesizes proteins, and produces organelles needed for its specific functions. For example, a muscle cell might produce additional proteins that aid in contraction, while a nerve cell might grow dendrites for better communication. Next comes the S phase, or synthesis phase. This stage is critical because it's when DNA replication occurs. DNA, or deoxyribonucleic acid, is the molecule that contains the instructions for building and maintaining an organism. During replication, the cell makes an exact copy of its DNA, so that each new cell will have a complete set of genetic instructions after division. The process of DNA replication is highly precise and involves several key steps. It begins with the unwinding of the DNA double helix, a structure resembling a twisted ladder. Special enzymes called helicases separate the two strands, creating a replication fork. Each strand serves as a template for the formation of a new complementary strand. Enzymes called DNA polymerases add nucleotides, adenine, thymine, cytosine, and guanine. Following the base pairing rules, adenine pairs with thymine, and cytosine pairs with guanine. The result is two identical DNA molecules, each with one original strand and one newly synthesized strand. This method, called semi-conservative replication, ensures that the genetic information is accurately preserved and passed on to the next generation of cells. After DNA replication, the cell enters the G2 phase, or GAP2. During this stage, the cell continues to grow and makes final preparations for division. It checks the replicated DNA for errors, ensuring that the genetic material is intact and ready to be distributed to the daughter cells. Once interphase is complete, the cell moves into the mitotic phase. The M phase consists of two major processes, mitosis and cytokinesis. Mitosis is the division of the cell's nucleus and its genetic material, while cytokinesis is the division of the cytoplasm, resulting in two separate daughter cells. Mitosis itself is a multi-step process with four main stages: prophase, metaphase, anaphase, telophase. During prophase, the DNA condenses into visible chromosomes and the nuclear envelope begins to break down. Spindle fibers form which will help separate the chromosomes. In metaphase, the chromosomes align in the center of the cell, forming what's known as the metaphase plate. This alignment ensures that each daughter cell will receive an identical set of chromosomes. During anaphase, the sister chromatids, identical copies of a chromosome, are pulled apart to opposite sides of the cell. This separation is crucial for maintaining the correct number of chromosomes in each daughter cell. Finally, in telophase, the chromosomes reach opposite ends of the cell and new nuclear envelopes form around them. The cell is now ready for the final step, cytokinesis. During cytokinesis, the cytoplasm divides, resulting in two identical daughter cells, each with a complete set of DNA and all the necessary organelles. The cell cycle doesn't just support growth, it also plays a key role in cell differentiation, the process by which cells become specialized for specific functions. For instance, during the development of a human embryo, cells differentiate into various types, such as muscle cells, nerve cells, and blood cells. Each type of cell has a unique structure and function, enabling the organism to perform complex tasks. Cell differentiation is closely tied to the cell cycle because it allows organisms to grow in size while also developing the specialized tissues and organs needed for survival. Imagine the cells in a leaf of a plant. Some differentiate into guard cells that regulate gas exchange, while others form the vascular tissue that transports water and nutrients. Without the cell cycle providing new cells for differentiation, such specialized structures couldn't form. It's also important to note that errors in the cell cycle can have serious consequences. If the cycle becomes unregulated, cells may divide uncontrollably, leading to conditions like cancer. Understanding the mechanisms of the cell cycle not only sheds light on how organisms grow but also helps scientists develop treatments for diseases caused by cell cycle disruptions. In conclusion, the cell cycle is the backbone of growth and development in living organisms. It enables the production of new cells, supports the process of differentiation, and ensures that the genetic information is accurately passed on. Through its carefully regulated stages from interphase to the mitotic phase, the cell cycle orchestrates the creation of life at a cellular level, contributing to the diversity and complexity of life on Earth.